Welcome back to Chapter 4, the three-dimensional structure of proteins. In this lecture, we will look at Part 4 of this chapter. Here, we'll continue our discussion on protein tertiary and quaternary structures. Specifically, we'll examine the structures of globular proteins. Globular proteins are different from fibrous proteins. In globular proteins, different segments of the polypeptide chain fold back on each other, generating a more compact shape than is seen in fibrous proteins. If you remember fibrous proteins, they don't fold back on themselves, right? They are combinations of secondary structures. We have seen two different examples, right? Keratin and collagen. Now, in globular proteins, the folding also provides the structural diversity necessary for proteins to carry out a wide array of biological functions. These are some of the characteristics of globular proteins. The structure of a globular protein is dictated by its sequence. Different proteins will have different amounts of secondary structure and different overall folds. Weak interactions drive folding. So, smaller proteins have fewer potential interaction partners. Now, this is crucial because weak forces or weak interactions are at play when it comes to protein folding. And you will have to recollect all those weak forces that you learned in Chapter 2. Small proteins typically are often stabilized by covalent bonds. And when I mean covalent bonds, I mean to say disulfide bonds between two cysteines. Shown here is an example of human serum albumin. This protein has 585 amino acid residues in a single chain. Now, given here are the approximate dimensions of a single polypeptide chain that it would have if it occurred entirely in a beta conformation, or as in an alpha helical conformation. Also shown is the size of the protein in its native globular form. So if it takes up the form of a globular protein, it doesn't need much space specifically, right? As compared to just taking up beta conformation, which means that this protein is made up of only beta sheets that is comprising of beta strands. It's such a long protein, right? Alpha helix, shorter than the beta conformation, but still longer than the native globular form. Now, this native globular form happens to be the most stable form of human serum albumin. And that is one of the reasons why globular proteins tend to fold. Shown in this slide is an example of a water-soluble globular protein. The name of this protein is myoglobin from the animal sperm whale. There is a reason why this specific protein was shown here. It is because this is the first globular protein that was crystallized, and the three-dimensional structure was obtained from a technique called X-ray diffraction studies. And this was done in 1950s. Myoglobin is a protein that is involved in carrying oxygen to muscle cells. Now, myoglobin is also particularly abundant in the muscles of diving mammals such as whales and seals. It is so abundant that the muscles of these animals are brown. Now, there are two things that you need to observe in this figure. The first one is the ribbon representation that is shown here on the left and a surface representation that is shown on the right. Now, ribbon representation allows you to identify the secondary structural elements. This is a tertiary structure of the protein which contains secondary structures, remember. Now, if you look closely, there are two different secondary structural elements. One is an alpha helix and another one is a beta turn. Between two helices, there is a turn. And that is what helps the protein to fold into a three-dimensional structure and a tertiary structure. The other thing that you need to notice is a red-colored molecule that is stuck to the protein. This is called as a heme group, H-E-M-E, a heme group. A heme group is what helps myoglobin to bind oxygen and store it and also carry it to muscle cells. Now, this is a prosthetic group that is attached to the protein. We will talk about this protein in detail when we talk about protein function. Now, about the surface representation. Surface representation allows you to visualize pockets in a protein. Now, the pockets are important because that tells you as to whether there are some molecules that are bound to the protein. These molecules could be ligands. So, two different representations. Another representation of a globular protein is by showing the individual amino acid side chains on the ribbon representation. Now here, the yellow sticks are the side chain of amino acids. Particularly, these are hydrophobic amino acids. Now, when you color it differently, you can see these side chains clearly. One of the reasons why this was shown is to show that these hydrophobic amino acid residues are in abundance in this protein, myoglobin, and they help this protein to fold. Remember, if it were to be a negatively charged side chain, it would be red in color, and if it were to be a positively charged side chain, it would be blue in color. And that color coding is also shown in the structural representation. Now, this is a space filling model. This is different from what we saw in the previous slide, which is a surface representation. Now, the space filling model is to emphasize the point and show the colors, saying that these are the regions where you have a hydrophobic residue. And these could be closer or away from the pockets that you want to see. To understand a complete three-dimensional structure, we need to analyze its folding patterns. But before that, we need to define two important terms that describe protein structural patterns or elements in a polypeptide chain. The first one is called as motifs or folds or supersecondary structures. A motif or a fold is a recognizable folding pattern involving two or more elements of secondary structure and the connections between them. A motif can be very simple, such as two elements of secondary structure folded against each other and represent only a small part of a protein. It could also be a very elaborate structure consisting of several secondary structure elements, like all alpha helix, all beta sheets, or a combination of both. Motifs can be found as recurring structures in numerous proteins. Proteins are made of different motifs folded together. Shown here are two different examples of motifs. On the left is a simple motif, that is a beta alpha beta loop. Here's where the protein starts. The N terminus, a beta strand followed by a loop, and then an alpha helix followed by a loop, and then a beta strand, C terminus. These two beta strands form a beta sheet. It can also be an elaborate motif called as a beta battle. A beta battle is a single domain of beta sheets. In fact, these beta sheets are formed by individual beta strands. So these are the examples of structural motifs. The second important term that we need to define is called as a domain. Large proteins will typically have multiple domains, each having several hundred amino acids folded into a discrete unit. But what is a domain? A domain is a part of the polypeptide that is independently stable or could undergo movement as a single entity with respect to the polypeptide. What does this mean? In a protein with two domains, for example, this protein that is shown here, right? Each domain may appear as a distinct globular lobe. So this looks different from this, but there are different amino acids that can have contacts between this domain and this domain. Now, different domains often have different functions or distinct functions, such as this domain may interact with small molecules like calcium in this case, and this domain may not necessarily have binding with calcium or other small molecules. Domains can have interaction with other proteins. For example, this domain may interact with proteins that this domain may not interact with. In addition, this domain could undergo movement without changing the conformation of this domain. That is the definition of a domain by itself. Like I said, some domains are functional and others are structural, purely structural, which means that their very existence is to define the structure of the protein. These are the common structural motifs, and some of the common structural motifs that we talked about in the previous slide could make up a domain as well. Having defined the two major structural patterns in a globular protein, let us now consider rules of folding for polypeptides. The first one is that hydrophobic forces drive protein folding. And the hydrophobic effect makes a large contribution to the stability of protein structures. Burial of hydrophobic amino acid side chains so as to exclude water requires at least 2 layers of secondary structural elements. This means that in a simple structure like an alpha beta loop, 2 separate layers are created, and the alpha helix remains as a separate layer versus the beta sheet remains as another separate layer. This is what it means. This is the first rule of protein folding. The second rule says that beta sheets and alpha helices rarely occur in the same layer. This is because the backbone of a polypeptide segment in the beta conformation cannot readily hydrogen bond to an alpha helix that is adjacent to it. If you look at it, a beta strand can hydrogen bond with another beta strand. but it rarely hydrogen bonds with an alpha helix. And that is the essence of the second rule. The third rule states that segments adjacent to each other in the amino acid sequence are usually stacked adjacent to each other in the folded structure. So if this amino acid and this amino acid are close in sequence, they are also close to each other in the tertiary structure. Distant segments of a polypeptide may come together in the tertiary structure. For example, if this amino acid, or for example, let's consider this amino acid. Now this amino acid and this amino acid may not be close in sequence, but in the tertiary structure or the folded structure, they can come close together in space. And that is what the third rule is. The fourth rule says that structures will almost never cross or form knots. And this applies for beta sheets. For example, when you consider two structures, a typical protein structure will exist in this form, where there is no crossover or knots. And this kind of a structure where there is a crossover and a knot happens is rarely observed in proteins. The beta conformation is most stable when the individual segments are twisted slightly in a right-handed sense, as shown here. This influences both the arrangement of beta sheets derived from the twisted segments and the path of the polypeptide connections between them. Two parallel beta strands, like these two, must be connected by a crossover strand. In principle, this crossover strand could have a right or a left-handed conformation. Now, how is this a right-handed conformation? Is if you were to look at, if you twist this, or if you rotate this 180 degrees, right? Then you could see that this starts from here and goes here. So it's going towards the right-hand side, and that's why it's a right-handed connection. Whereas this, it's going from here to here, and that's why it's a left-handed connection. Right-handed connections tend to be shorter than left-handed connections and tend to bend through smaller angles, making them easier to form. The twisting of beta sheets also leads to a characteristic twisting of the structure formed by many segments together, as seen in a beta barrel that we saw previously. So among right-handed and left-handed, this is almost always the case in proteins. Following these rules, a series of beta-alpha-beta loop, which is a smaller motif, can combine together to form a larger motif like alpha-beta barrel. Now, beta strands in this alpha-beta barrel creates a particularly stable and common motif. In this structure, each parallel beta segment is attached to its neighbor by an alpha helical segment. All connections in this protein are right-handed. The alpha-beta barrel is found in many enzymes, often with a binding site in the middle, and that is for a substrate or a cofactor or a prosthetic group to bind. And the domains with similar folding patterns are said to have the same motif, even though their consistent alpha helices and beta sheets may differ in length. At this point, I would like to introduce two different techniques that are used to determine the three-dimensional structure of a protein. The first one is called as X-ray crystallography. These are the steps needed to conduct this experiment. First, you need a pure protein. For that, you need to purify recombinant protein. Next, you need to crystallize the purified protein. Once you have a crystal, you shine X-ray through this crystal, and this crystal will diffract X-rays, creating diffraction data. Calculate electron density from the collected diffraction data and fit known amino acid residues into this density. Now, what I meant by these three lines will be clearer in the next slide. There are certain pros and cons for this method. The pros is that it can crystallize proteins up to 150 kilodalton, and you can obtain the structure of these crystallized protein. This is a well-established methodology. However, the cons are that it's extremely difficult to crystallize membrane proteins, and you cannot resolve hydrogens or C hydrogens. Difficult for higher molecular weight proteins. Proteins that are above 150 kilodaltons, it's extremely difficult. Now, to circumvent the second and the third issues, there are two other methods. Neutron diffraction is used if one needs to see hydrogens in proteins. Now, to determine structures of proteins above 150 kilodaltons, another technique called as cryoelectron microscopy is used. Here is a little more detailed explanation as to what I said in the previous slide. Once you shine X-rays through a protein crystal, you will obtain an X-ray diffraction pattern. Now, data extracted from the diffraction patterns are used to calculate a three-dimensional electron density map, which looks like this. Usually, Fourier transform is performed. Regions of greatest electron density reveal the location of atomic nuclei, and this information is used to piece together the final structure. Here, the structure of heme, H-E-M-E, the one that I talked to you about in the previous slide, is modeled into the electron density map. And this will be found in the interior of the protein. The same way, all these proteins are modeled from the electron density map. The second technique is called biomolecular NMR, or nuclear magnetic resonance. Those who have taken organic may remember as to what NMR is, right? Now, the steps needed are very similar. You need a pure protein. Now, instead of crystallizing the protein, you need to dissolve the protein in a buffer and then collect NMR data, assign NMR signals, and then calculate the structure. The pros are no need to crystallize the protein. That is a big positive. It can see many hydrogens. And in addition to neutron diffraction, where you can see hydrogens from a crystal, this technique, which you can see hydrogens from solutions, is employed. The limitations is difficult for insoluble proteins, and it works best with small proteins. So anything above 30 to 40 kilodaltons, it becomes increasingly difficult to resolve hydrogens and to determine the three-dimensional structure. Again, shown here is an example. where two-dimensional NMR is used to generate a three-dimensional structure of myoglobin. Now, the diagonal, as shown here, in a two-dimensional NMR, where one dimension is a proton chemical shift, the other dimension is also another proton chemical shift. The diagonal is equivalent to a one-dimensional spectrum. The off-diagonal peaks that are shown here are what are called as NOE signals, or nuclear Overhauser effect signals, generated by close range interactions of protons that may generate signals quite distant in the one-dimensional spectrum. So two such interactions are shown here. Now, from this that is being resolved, you can figure out the structure, the three-dimensional structure of a protein, and that is because this spectra gives the interactions of atoms in space, and by resolving it, one obtains a three-dimensional structure of a protein. Proteins can be classified by structural motifs. Only about 1,000 structural motifs or domains have been identified in natural proteins. Their organization and arrangement, however, differ. Protein folds are much more conserved than protein sequences. Remember, protein fold is a three-dimensional structure, a tertiary structure. Protein folds organized into families based on similar sequence, structure, and function. Superfamilies, on the other hand, are collections of families with likely shared evolutionary ancestry. The three-dimensional structures of proteins determined by X-ray crystallography, or NMR, or cryoelectron microscopy are deposited in a repository called as Protein Data Bank, or PDB. Now, in this data bank, proteins are usually classified based on folds and subfolds. The PDB identifier is a code that is a unique code that gives you the structure of that specific protein. And then the fold, superfamily, family, protein, and species are various different other identifiers. For example, in an all alpha family where everything is alpha helix in these tertiary structures, as you can see from here, serum albumin, and it belongs to humans, is an all alpha structure. The other all alpha structure that we saw is myoglobin, remember. All beta proteins such as UDP N-acetylglucosamine acetyltransferase belongs to a single-stranded left-handed beta helix fold, as shown here. This is from E. coli, a bacterium. Another protein like collagenase 3 carboxy terminal domain like protein has a four-bladed beta propeller like fold. It's from humans. CD8 is another protein that has an immunoglobulin like beta sandwich domain. So these are three different folds, and these belong to all beta family. Continuing our discussion here, alpha slash beta family, or alpha beta barrel kind of families are plenty. For example, alcohol dehydrogenase is another protein that belongs to NADP binding Ross-Manfold domains, or another protein like ATP dependent phosphofructokinase belonging to the phosphofructokinase fold. Now, for this protein, there are two different domains here. So these are some of the classic examples. And the takeaway from this is that three different things: motifs, domains, and folds. These are the things that make up globular proteins. Quaternary structures result from interactions between the subunits of multi-subunit proteins or large protein assemblies. Proteins will often interact with other proteins in complexes. This is called as oligomerization. Oligomers can be homooligomers or heterooligomers. Hemoglobin is a classic example. It's a homotetramer. It was the first oligomeric structure that was ever solved. Oligomers with repeating subunits are often arranged symmetrically. Remember, myoglobin was the first structure for which the three-dimensional structure was identified. And myoglobin happened to be a monomer, whereas hemoglobin is a homotetramer. We'll learn about myoglobin and hemoglobin later in protein function chapter. Now, in this diagram that is shown here, a ribbon representation of hemoglobin shows several secondary structural elements of the structure and the positioning of all the heme prosthetic groups. On the right is a surface representation or a surface model showing the pockets in which the heme prosthetic groups are bound and helps to visualize subunit packing. And you can see that there are four different colors: a blue colored, a light blue colored, white, and gray, depicting that there are four subunits because it's a homotetramer.